right, so I welcome you as for the first talk into this session, on new methods in nanomedicine. And I'm always delighted, to have the opportunity to speak here, because people who talk about simulations are not exactly overrepresented, let's say, in this conference. So I hope in the next 15 minutes I will have the opportunity, to convince you that some of our endeavors are sort of worthwhile. Right? This conference is getting more and more convenient over the years. They now upload your presentations, which basically would probably run without me staying here. And so they now invented this video thing to make sure that the speakers actually show up for the talks. Okay, so with this, I would like to introduce you into my talk, which is simulation in nanomedicine. I'm come from a modeling group, and of course there's lots of potential things that you could model at various length and time scales. And the thing which I'm going to talk about today is molecular simulation. And in that, of course, we sort of profit from two sort of converging trends, right? The first one is that mostly due to the growth in computational capacities, the types of things that we can simulate has gotten better and better. Also, there was a lot of method development over the years, right? And on the other side, the things that people have been interested in medicine, and in particular in nanomedicine, have gotten smaller and smaller, right? And so if you have these two trends, then it is just a question of time when these two lines that are schematically illustrated here intersect, right? And at this point, a new methodology starts to have some impact in this field, in this case, nanomedicine. And the types of simulation that we've been working on in my group for the last 15 years or so are molecular simulations, which now permit you to essentially take a biomacromolecule. We are still kind of quite limited in the size of what we can do, to the order of 100 amino acids or sometimes a little bit more, and completely characterize what it's doing, if it's left by itself, or also to a certain degree if you modify its environment, or have it interact with other things, right? And on this basis, you now have kind of an atomic scale movie of what's happening in your system, right? And that helps you to characterize particular key interactions, right? And replace a certain set of experiments, right, and maybe even suggest to experimentalists what they could do in order to modify the system. And I will give you at the end of the talk, if time permits, a short example where you have one of these questions that is at the moment basically impossible to address experimentally, where simulation can be really useful. Right? So, one of the key things, rather, going beyond modeling the molecule by itself, is to actually model, the interactions with small molecule ligands, with surfaces, with peptides, with membranes, and with nanoparticles. And it is quite easy to extend the technology that I've shown sort of in the previous movie to this type of thing, right? And the first thing you can then do is essentially, in silico screening, where you start now with a large database. You can download databases with up to 8 million compounds. That is comparable to what large pharma companies have for wet screening, right? And you can screen those, right? Identify the first 10 or so hits, right? Talk to some medicinal chemist, what may or may not be toxic, synthesize the top 10, and get sufficiently lucky that you're actually able, to identify on the basis of in silico screening alone nanomolar ligands, which in this case affect the affinity of heparin, right, and act as an anticoagulant, right? So that's one of the things that you can do. The second thing we were interested in, for instance, is to look at toxicity effects, right, for small molecule ligands. And later I will talk about nanoparticles, and one of the key channels that you look for this is the Herc channel. That is one of the standards that you have to test against if you want to develop a new drug today. And in doing that, we were able to look at a certain set of small molecule ligands. The problem is that there is no really a crystal structure for the Herc channel, right? And so you have to make up a homology model based on data for similar channels, and that you can then validate by essentially reproducing experimental results in your in silico screening, right? And so that we have done, and then we were able to identify certain key interactions, right, and look at mutations in the channel, right, that were able to significantly affect the activity of known drugs interacting with the channel. And this was done in cooperation with the group of Dirk Thomas at the cardiology in Heidelberg. Okay, and then we went off and said, now we have the channel and we figured out some protocol to reconstitute it in various environments. And we wanted to look at the toxicity of small gold nanoparticles, right? And that turned out to be some rather painful endeavor because we were surprised step after step at what was going on. And again, it was in the end simulations which gave some hints what was going on. And to make the brief story short, I think you've heard, in this conference and other locations over and over, that when you look at the, toxicity of nanoparticles, there's two key constituents, right? And the key constituents that you're typically told you should look at is the surface of the naked particle, right? But then often it comes with a ligand shell, right? And then you look at the surface of the ligand shell, right? And one of these two things, is typically responsible for the interactions with the environment and the toxicity of the particle, right? And so that is basically the thing that we tried to do. We took the gold nanoparticles; they fall apart immediately if you don't coat them with some ligand shell, and we put them, in various concentration. And we found that, in vitro there was an irreversible blocking of the channel, right, for nanoparticles coated with a certain ligand, while if you just change the phosphate groups that anchored the ligand on the nanoparticle, the effect was completely switched off, right? And so what we then went off is, we went back to the model of the Herc channel that we already had from the previous study, and we tried to dock the gold nanoparticle into the cavity, and we found out that this would never work, right? First of all, the nanoparticle with the ligand shell would not possibly fit into the cavity of the channel, right? And secondly, the interactions between the nanoparticle and the coated nanoparticle and the channel were repulsive, right? So if I'd shown you the movie of this, you would put the nanoparticle in front of the channel, and the nanoparticle would just go whoosh, right? And after you've looked at this for 10 times, and made sure that your student hadn't messed up the simulation, we thought that there must be some sort of an effect, right? So what happens here is actually ligand replacement, right? We hypothesized that the ligands would come off the gold nanoparticle, which turns the gold nanoparticle from something that's hydrophilic to something that's hydrophobic, right? And the hydrophobic nanoparticle would then block the channel. Right? And by modulating the strength with which the ligand binds to the nanoparticle, you can either have or not have this effect, right? This was then validated in vivo, where we took oocytes and tried to block them with a nanoparticle where the ligand would not come off, and the oocytes didn't care at all, right? And if you take the same oocytes and you put a nanoparticle where you can where you've replaced the ligand, you would block the channel, right? So in order to figure out what on earth is going on, or to validate the scheme, two of my students, who are actually in theory, went off to experiment, and they conducted these black lipid membrane experiments where you have a lipid bilayer in some small membrane, and then you put some current, and now you put the nanoparticles in one size, and you observe what you do, right? And so it turns out that as we know, when we vary the size of the nanoparticles, right, there seems to be a critical size below which nothing happens, right? And that size has something to do with the size of the membrane, right? So therefore, if the nanoparticle has a sufficient size to span the lipid membrane, then it embeds, right? And again, it embeds by the same mechanism that is relevant for the Herc channel that I've talked about before. So the nanoparticles, as far as we can tell today, do not traverse the membrane, but when they reach a critical size, they embed, right? And that then permits an ion-selective channel, ion-selective current, right, to permeate the membrane, and that is what you pick up as a signal in this type of experiments. Okay, and now I want to give you sort of as a last example a brief overview of a project where experiment pretty much has struggled a lot, and that is about the protein translocation of fully folded proteins out of the cell. Most of you know that their cells have some way to export proteins, right? Because they need that, because some proteins are relevant, in the extracellular environment. The pathway that you typically read about in biology books is the SEC pathway, which has a very small pore, right? So you take your peptide protein that you want to export, you tag on a signal peptide, right? That then unfolds the protein and channels it kind of through the pathway where it folds again, through the channel where it folds again, right? Now this doesn't work for a lot of enzymes which either bind cofactors, which are not available in the extracellular environment, or which need a chaperone to fold, right? And so therefore, there's a second channel, which as of yet is still largely unresolved, right? That works for me in a kind of fascinating way, right? You punch a hole in the membrane, which has exactly the size of the protein that you want to translocate, right? You channel the protein through that in a fully folded fashion, right? And then you close the pore, right? Because if you kept the pore open, then the cell would be dead within minutes, right? Okay, and the way this works is largely not understood, because this TAT channel translocates proteins of vastly different sizes, right? So it's not a single type of pore which does it, right? So the pore must be kind of made, tailor-made to the size of the protein that you want to translocate, right? Okay, now, since this happens in the membrane, and it is small peptides, TAT R, B, and C is actually a large protein, so TAT A and B are the two peptides which make up the membrane, right? There is today no crystal structure and very little information beyond cryo-EM what's going on there, right? But our experimental colleagues figured out a scheme by comparing lots of sequences across different species, what's going on, right? So if you look at these peptides, then you have a hydrophobic transmembrane sequence, which is something that inserts into the membrane, right? And then you have something that you cannot make much sense of, because it's a relatively charged, helical thing, and this you can get from solid-state NMR, which you would say sticks into the solution, because you cannot possibly put it into the hydrophobic membrane, right? But then, the experimentalists noticed that there is a complementarity of charged side chains, right, that would enable you to kind of glue these things together, right? And that might be a mechanism, right, that would give you a like a palisade, right, around sort of a medieval village, right, that would have a hydrophobic exterior made out of this stuff, right, and a hydrophilic interior made out of this stuff, right, that would be, the hydrophobic environment that permits the protein to translocate, right? Okay, but nobody knew, based on this picture, how to build up this thing, and we could make simulations, which I show you here, right, that gave the first sort of atomistically resolved picture of this complex, right? You can compare it with the cryo-EM pictures, and to the degree of the resolution that we have, we can say that they reasonably agree, right? And this model, of course, by itself doesn't prove anything, right? And that was also the opinion of the editors of Cell. But it suggests that the experiments, right, of what you can do in order to mutate specific residues on these amino acids in order to break the salt bridges that would hold this complex together, right? Which we managed to do, right? So we've broken it, right? And then to suggest new mutations that would reconstitute different pairs, right? So that you could rebuild it, right? And by performing these experiments, which was a lot of work done in the group of Anne Ulrich at KIT, we were able to basically demonstrate that this thing actually exists, right? And that we can now manipulate sort of when and how it is formed, right? And with that, I'm actually coming to the end. The chairman is smiling. And I would thank our experimental co-workers for all of this work. So the work on the Herc channel was done together with the groups of Simon and Jan and Dichent in Aachen, who make and characterize these nanoparticles in vitro, with the group of Thomas at the cardiology in Heidelberg. The work on the translocation was done with the Ulrich group and the Rogerone group. And they are sort of the biology. I didn't talk today about human developmental disorders, and the antithrombin work, which I touched upon before, was done together with these groups in Murcia in Spain. Thank you very much for your attention. Yes. Yes.
They were naked or coated?
Well, that's the key thing, right? They were coated when we put them in, right? Because in solution, they would immediately disintegrate if they're not coated, right? But depending on which environment they see, they may shed the ligands, and then they're no longer coated. Okay? And that was sort of one of the key things, right? So, if you want to put them in the Herc channel, right, then they must lose their coating, right? And depending, by modulating the ligand strength, the binding onto the nanoparticle, right, you can switch this effect in various environments.
I have another question, if I may.
Please.
The protein ligand interactions that
Yes.
you get into the nanomolar part. We try to do the same with decapeptides and proteins. We didn't go further than millimolar or micromolar. If you very poor binding, do you have any reason for us to hang on?
Sorry?
I think the question was, in the screening, did you find anything really high potency? In my experience is you never will, but...
Yeah, no, I mean, right, sort of, yeah. I mean, sort of, I don't think that without sort of a major improvement round, you become sort of into the low nanomolar range. With screening, you can get, if you get lucky, into the high nanomolar range, but even then you get lucky. Typically, you end up in the medium micromolar, right, and then you go into ligand optimization. Yeah.
Lower millimolar and high micro. I wonder what was the reasons for it. Solubility in water of both partners.
Sorry?
The reasons for why it's impossible to find a good
it's
peptide.
because it's so small.
Yeah,
it's so small, you haven't got enough
I mean,
space chemically to get a nanomolar interaction.
We had 20,000 mutations.
Yes, you could have that, but you've still got limited information in that small chemical entity. If you increase it,